think what I'll try to do is just very briefly summarize or sort of touch on some of the things that are going on in cancer research in general, and then focus a little bit more on blood cancers, and then focus down on acute leukemia. And I think one of the things we'll begin to understand as we go through this is that there are a lot of similarities among the blood cancers, the so-called blood cancers: acute leukemia, chronic leukemia, multiple myeloma, lymphoma, Hodgkin's lymphoma, and non-Hodgkin's lymphoma both. They share some features as we're beginning to understand them a little bit better. They share both biologic and clinical features that sort of tie some of the research together that we never really understood before. First, I just want to say that the work of this organization, the Leukemia Research Foundation, and organizations like it are extremely important now, especially as the funding from national, federal organizations, National Institute of Health most prominently, has been reduced drastically over the past maybe 10, 15 years or so. We're at a point of funding for research where no more than about 7% of all the grants that are applied for are actually funded. That's a number that's never been lower, at least in my time doing this, and I don't think that it's actually ever been lower. That means that 93% of the grants that are submitted to the NIH are turned down, are not funded. What that has resulted in is sort of a drain of people that are interested in attempting to do research in cancer or in other areas, in cardiology, and this goes across all fields of medicine. So when we think about how to fund where we want to put our federal dollars, we think about defense. I think of cancer research as one of the major tenets, major parts of national defense that we have. And I think this should be clearly a focus for our efforts, for fundraising, for politics, to try and get people to understand the importance of funding for cancer research. So it's really the work of organizations like this one that's kept things going, that's enabled people to begin doing research in these areas. So one of the major findings, a number of findings that have led to changes in the way we approach this and other diseases has been the identification of important genes that have existed but we never knew about. We have now methods to identify genes which are abnormal in certain diseases and have become abnormal in certain diseases. That doesn't mean that these are heritable genes. These are genes that have acquired certain abnormalities that lead to diseases like, for example, acute leukemia. And specifically in acute leukemia, we're beginning to identify certain genes which predict either a better outcome or a worse outcome, number one, so that we can treat patients not all the same. We may have two patients with acute myeloid leukemia who under the microscope look identical in terms of their disease, but who have very different molecular characteristics. When you look at the genes that are involved, the chromosomes and the DNA that's abnormal, they're very different from one patient to the next. So, number one, we can recognize differences in clinical behavior by looking at these genes, but number two, we can also begin to identify drugs or molecules that block or reverse the genetic abnormality. And that's been a focus of cancer research, I'd say probably over the past 5 to 10 years. And specifically in acute leukemia, there are genes called FLT genes, FLT genes, FLT 1, 2, 3. There are a number of genes which have been identified as being important in acute myeloid leukemia, but we now have specific inhibitors of these genes that can reverse the process. A good example of that occurs in a disease that's related to acute myeloid leukemia called chronic myeloid leukemia or chronic myelogenous leukemia, where people have been able to identify the specific abnormality that exists that causes the disease, and as a consequence, there are drugs, and you may have heard of these drugs like Gleevec, as a prime example, which are pills that people take on a daily basis that totally reverse the genetic abnormality, the molecular abnormality of chronic myelogenous leukemia. It has really changed the whole face of what was once a uniformly fatal disease into one that is either a curable disease or certainly a chronic disease where people can live normal lives. The same kind of work is now being done in acute myeloid leukemia, where there are pills that are being developed, oral agents which are being developed that reverse the whole abnormality that causes these disorders. In chronic leukemias, chronic lymphoid leukemia, which is probably the most common form of leukemia in adults, there are totally new approaches to the treatment of these diseases that depend on recognizing either abnormal molecules or abnormal proteins on the surface of the cells that have led to major changes in the way these diseases are treated. We're now working with a group of drugs called BTK inhibitors. These are oral drugs which are not yet available for routine use, but are being used in clinical trials across this country and across the world. And some of the early results of those trials have been phenomenal. They have really changed, I think are beginning to change, the face of how we approach these diseases. And I think it's going to be probably not more than 1 or maybe 2 years before these drugs will be approved for routine use by the FDA, and probably will begin to enter first-line treatment for a variety of chronic leukemias. In the lymphomas, both Hodgkin's and non-Hodgkin's lymphoma, there are similarly new agents and new approaches that are being tested, that are being evaluated both in laboratories first and then in clinical trials, looking for new ways to treat those diseases as well. The other area is prevention. We unfortunately haven't learned yet how best to prevent these diseases because we haven't yet learned what causes most of them. We have a clue. We think that some may involve certain viruses, for example, in the lymphomas. And so blocking those viruses in some way may be a way to prevent these diseases. But as we begin to learn more about the natural progression, the molecular pathways that are involved in causing these diseases, we can begin to think about ways to actually prevent them. I think we're probably a little farther away from a prevention than we are for long remissions or cure in patients who develop these diseases. So I think the other area that we're involved in, both in leukemia and in lymphoma, is the use of something called nanotechnology. We are now using particles, nanoparticles that can't be seen except by special electron microscopes as a way of carrying certain drugs directly to the leukemia cells without causing toxicity to normal cells. So some of the things you just heard about, the infections that we see associated with treatment of acute leukemia, may not be a problem in the very near future as we target just the abnormal cell. And so I think that with the kind of research that this organization is funding and that you, by your presence here, are fostering, we're going to make, I believe, some very major strides in the next 5 to 10 years in all of the blood cancers, the acute leukemias, chronic leukemias, lymphomas, and myelomas. So I wanted to thank all of you for your efforts on the behalf of John and Barbara, and on behalf of all of those who are touched by these diseases. My mother died from acute leukemia, and so I have an understanding, I think perhaps with John, on how this can affect families and how this can affect all of us. And again, the work of this organization and your work is so very greatly appreciated. Thank you very much.